right, folks, it's time for us to get started here. One more time, I'll remind you that 2 weeks from today, you'll want to get here to this room early because it's going to be packed with people who are just eager to take the first exam for this class. All right, so 2 weeks from today. The exam's going to look an awful lot like the ones that you'll find on the pilot page for the course and in the back of the lecture notes. I'm anticipating 45 multiple choice questions. You should bring a pencil, right, because you're going to be filling out little bubbles on Scantron forms to get them graded, right? There are no essay questions or anything like that. So it's not too early to start thinking about that first test. Take a look at those old exams. That should help you quite a bit, all right? Now, I have your attention, right? Let's start talking about some of the biology that is going to be the topic for today. Finally, I get to move on to the really cool parts of molecular biology, essentially. One of my favorite things to talk about in this class. Just to give you guys a little bit of truth in advertising, I suppose, from my perspective here. The things I'm going to talk with you about today are essentially proteins and nucleic acids, DNA and RNA, right? And these are topics that really get me excited. They get my blood pumping. And that's been the case for quite some time. I think it's my interest in how proteins do what they do and how DNA in turn tells proteins to do what they do. It's been that interest that has sort of been what's been driving me for my own professional career for the last 20 or so years. I've written a book, a textbook, a widely used undergraduate textbook on bioinformatics. The title of the book is Fundamental Concepts of Bioinformatics. You can tell it's got all the makings of a New York Times bestseller, right? With a title like that, how can you lose? But that book is used in undergraduate courses all over the world. There are many different languages into which it's been translated, and it's something that I use in a course that I teach here at Wright State often. Typically in the spring, one of the classes that I'll teach is a bioinformatics course. What I'm going to talk with you about today is just a thumbnail sketch of what we do in that bioinformatics course, where we take the sequence of genes and we take the sequence of proteins and we use that information to make predictions about what sort of structures a molecule will fold up into, and then in turn what kind of chemical reactivity it's going to have. So again, today I'm just going to give a glancing blow to some of the stuff that has fascinated me enough that I had the it gave me the stamina to actually write a book about it, right? You got to have some pretty serious interest in a topic to sit down and write out a whole treatise about it. And so that's what I want to talk with you about today. If what I'm saying excites you the way it excites me, we got stuff for you to do here at Wright State. There are courses you can take. There are books that I can sell you, right? There's all sorts of things we can do to help round out your interest in this if you have the same interest that I do. All right, but so that's what's on the schedule for today. This should be a lot of fun. Let me remind you where we've been. I seem to do this every lecture. It gets me warmed up if nothing else, but maybe gives a little continuity between one lecture and the next. When we met on Tuesday, I was talking with you for the first time about biopolymers, about stringing together these small things that we call monomers to make larger, more complicated molecules. The first one that we talked about, the first one that I told you about, were the carbohydrates, where the monomers are single sugars, monosaccharides, that can get strung together into long chains to make starches and amylose and things of that nature. Those carbohydrates are essentially used for energy storage, but they can also play some important roles as a functional sort of molecule, structural. They give rise to some rigid structures if you put them together in a particular way. And plants and insects use them to some advantage by playing that trick. Humans, mammals, other animals, not quite so much. So carbohydrates, their primary job is energy storage. You could say energy transfer. It's relatively easy to store energy in their covalent nonpolar bonds and to deliver that energy from one place in the organism to another. That's their role. A main point, I hate to say it's a main point, maybe I should say it's one of the points that I made along the way is that, like with all the polymers, when you make a longer chain of carbohydrates, you do that with a condensation reaction. So a water molecule gets kicked out of as you link together two monomers, a water molecule leaves. You break those chains, typically from the end in, by adding a water molecule. So that's a hydrolysis, breaking of the bond by water, and you link them together with condensation reactions. So those are carbohydrates. Then I did a glancing blow at the second of the four big players in terms of biopolymers, and those were the lipids. Lipids are only, I think you have to fight a little bit to say that lipids are in the same league as these other three biopolymers that we'll be talking about. Carbohydrates, the length of those chains, the length of those polymers, there is effectively no bound. There is no practical limit to how long the carbohydrate chain could be in one of those biopolymers. And the same is true for proteins and nucleic acids. Proteins, a typical protein will be about 200 amino acids long. That's where we're going in just a couple minutes. A typical nucleic acid, a chromosome, can be millions, tens of millions, even hundreds of millions of nucleotides long. Lipids don't quite fit the bill the same way as those other three biopolymers do. They tend to be triacylglycerides. When we're talking about lipid polymers, we're usually, in a biological sense, talking about triacylglycerides, meaning that there are three fatty acids that get held together on one glycerol. Now you put those fatty acids on in the same sort of condensation reaction, so it meets that criteria of being a polymer. You pull those fatty acids off with hydrolysis reactions, so it's a polymer in that sense. But you don't get these really long, crazy, million monomer long chains. They tend to be clustered in groups of three. In terms of a function for the lipids, they are primarily long-term energy storage, lots and lots of nonpolar covalent bonds. That's pretty much all that you see when you're looking at the structure of a fatty acid, of a lipid. But living things have also put them to work in some interesting structural ways. And so I skimmed over this. I hit this really quickly while people were starting to put papers away and bundling up to leave on Tuesday. And I wanted to just one more time draw your attention to this particular structure. This is another kind of lipid. This isn't a triacylglyceride. This is a phospholipid, where here what we've got is the glycerol is still there to hold the fatty acids together. So this 3-carbon molecule is a glycerol. And you can see there are 2 fatty acid chains dangling off of it. But instead of a 3rd fatty acid chain, what we have on these phospholipids is a phosphate group. So 2 of the carbons in the glycerol are attachment points for fatty acids. 1 of them is an attachment point for a phosphate group. When I say phosphate group, I hope that you guys I hope you get conditioned to pretty much immediately translate that to, phosphate group. What does it? Negative charge, right? That's all that matters about a phosphate group. There's nothing subtle. There's nothing more. There's no secondary consideration when I say phosphate group. When I say phosphate group, I hope that conjures up in your mind a mental picture of an intense, localized negative charge, right? And so these phospholipids have the hydrophobic tails from their fatty acids on one end, and then also associated with this molecule is this phosphate group that gives to it a really intense, very focused, very localized negative charge. I used the word schizophrenic at the end of lecture on Tuesday, and people chuckled about that a little bit, right? I mean that really seriously. You know, in terms of behavior of this molecule, schizophrenic is the right word to describe it. One end of this molecule, the fatty acid end, has absolutely no basis for interacting with water. Nothing but nonpolar covalent bonds. Carbon to carbon, carbon to hydrogen bonds all over the place. In a word, it's hydrophobic. It's afraid of water. It actively tries to get out of the way of water. It's uncomfortable near water. Water wants nothing to do with it. No matter how you're coming at it, that has a very specific kind of behavior because of its chemical structure. And exactly the opposite would be said about a phosphate group, that other part of this phospholipid. So in one molecule, it has two very distinctly different kinds of behavior. One that is profoundly hydrophobic, and another part that is profoundly, and I don't use the word profoundly here lightly, I mean it really seriously, profoundly hydrophilic, right? So at the same time, this one molecule, two sides of its face, right, it's just completely opposite in terms of its behavior. Those are phospholipids. Those are the principal component of all living things' cell membranes. And so on Tuesday of next week, we're going to start talking about how all this stuff comes together, all these biopolymers, how they come together to make cells. One of the first things that I'll talk with you about when we're talking about cells is these phospholipid bilayer membranes, the cell membranes that define the boundary between what's the outside of the cell and what's the inside of the cell. Those phospholipid bilayers are made out of these molecules. So here, these fatty acids, while there's a lot of energy that's being stored in their covalent bonds, that's not really the role, that's not really what living things are putting them to use for. They're playing an important structural role. So lipids, great for long-term energy storage, and then also play this really important role for making membranes. Here's a picture of a single phospholipid. Here's a whole party full of phospholipids. Same general sort of schematic, right, but now a little bit wider view. The hydrophobic heads, those are those phosphate groups attached directly to the glycerol. The the hydrophobic tails, those are the fatty acid chains, the two of them that are dangling down. A stable way to arrange these molecules, in fact, the most stable and perhaps the only stable way to arrange these molecules is the way that you see them portrayed up here. You can have the hydrophobic tails come together from two different directions to sort of provide each other some cover, a water exclusion zone, if you like. And on either end of that, you've got the layer of the phosphate groups with their really intense negative charge that water loves to interact with and makes those phosphate groups feel happier. Right? And so that structure, again, is a phospholipid bilayer, because there's two, in the layer. Right? There's a layer, so it's a bilayer. And again, that's what cell membranes look like. And we'll talk about those a fair bit more on Tuesday of next week. All right, so that's the outline from Tuesday's lecture. Today, we're moving on, and let's get to the good stuff now. Let's start talking about this other biopolymer called proteins. Okay, the monomer that is the relevant thing to talk about when we're talking about proteins are amino acids. All amino acids have this basic structural formula. Okay, so this is the monomer that we're going to string together to make, through condensation reactions, to be able to make a protein, or if you like, a polypeptide. Here's the lay of the land. Every amino acid is going to have this structure. They will all have an amino group. They will all have a carboxyl group. They will all have a central carbon. Technically, people will refer to it as the alpha carbon. And off of that alpha carbon, or that central carbon, will be a hydrogen and something else. Right? So if you look at that chemical structure, right, it's obeying the rules. Each carbon has 4 covalent bonds. Each hydrogen has 1 covalent bond. Each oxygen has 2. The nitrogen's got 3. Everything's cool that way. All amino acids have this basic structure, and the way that one amino acid differs from another is what's going on where we have R on this particular image. R is short for reactive group. Okay? That's where the letter R comes from in this context. And here's the thing. There are 20 different commonly used amino acids by all living things on this planet. Each of those 20 different amino acids differ from each other only in the sense of what that R is. Okay, so there are 20 different things you could plug in place of that R. Each of those corresponds to a different amino acid. Okay? I was trying to get you guys excited about, and I was using what I thought were very colorful terms when I was talking about using a palette of different chemical groups. Right? You have 7 different basic colors and the 7 different functional groups that living things turn to time and again. Right? I was trying to conjure up this sort of artistic way of synthesizing or playing games with the chemistry that living things do. Well, here we've got a palette with more shades of gray. We're using those 7 functional groups that are commonly used, but we're arranging them in different configurations in such a way that we get now 20 different shades that we can paint with, 20 different colors or hues, if you prefer. All right? And those 20 different combinations of the functional groups that give rise to these R groups give us the ability then to make molecules, polymers, that have different kinds of chemistry associated with them along the length of the polymer. All right? And it ends up letting living things do phenomenally cool stuff. Right? Living things take absolute full advantage of painting with those 20 different hues by stringing together amino acids with those different chemical groups. 20 different chemical groups, 20 different R groups, 20 different amino acids, all synonymous, all one and the same. So here's the basic structure, right? Every amino acid has a central carbon, an amino group, a carboxyl group, and a hydrogen. They differ in these R groups. Here are all the 20 different R groups attached to their amino acids. I want to draw your attention to a couple things. First, you probably can't make out what's going on this slide, especially if you're in the back of the room. It's too fine a print. Let me put you at ease a little bit. You don't have to worry too much about the absolute fine details of these structures. I'll tell you this. There are other people who teach this Bio 1120 class. Somebody else will teach the trailer section for this class in the spring. Most of the other people who teach this class insist that the class memorize the structure of each of the 20 amino acids, okay, those R groups. I'm not that guy. And I'm not going to make you memorize the structure of those R groups. I'll tell you, if you go on to take a sophomore level biology class or anything beyond, it's going to be assumed that you've memorized them. And I'll tell you from my own personal experience that it gets easier to memorize them each time you memorize them. Right? So if you're thinking you're going to take some more of these classes, maybe set aside, I think, an hour or 2 should suffice to let you memorize all of these stinking R group structures. My experience was it took me about an hour or 2 the first time. It took me half as long the second time. It took me half as long of that the time after that. I'm going to be completely honest with you. I'm going to struggle right now with coming up with a couple of those structures, but you give me 2 minutes glancing at that slide and I'll have them because I've memorized them so many times before. But if you asked me, I'll tell you what, if you challenged one of the biology faculty, any of the biology faculty, myself included, okay, drop and give me 20, right, go up to, but no, drop and here's a pencil and a paper, write out for me arginine, right? That's one of the 20 amino acids. They'll come up with some clever excuse about, oh, gee, this is an inconvenient time, I'm late for a meeting, that person there has an important question for me, right? But they won't be able to do it. It's going to be a really rare individual who would be able to sketch those structures out for you. So what's the point? Well, I got a point. I don't care if you memorize the structures or not. But I want you to be able to look at those structures. This is a consistent theme I have now, right? I want you to be able to look at the structure, and I want you to be able to tell at a glance what it is that's important, what it is, what kind of behavior a molecule will have that has this particular R group associated with it, right? And I got some reasonably good news for you. I told you there's 20 different hues in this palette that you can work with to make a protein that can do all sorts of amazing things. But there are really only 4 basic colors in your palette. There's like 3 or 4 different shades of each of those, 5 or 6 for a few of them. There are a number of different shades, but there are 4 basic color groups that I think you want to keep your eye on here. There are 4 different classes, 4 different kinds of amino acids because of the differences in their R groups. There are amino acids. Here, I'm going to rattle them off. I'm going to count them for you on my fingers here. There are 4 different classes of amino acids. There are the non-polar or the hydrophobic amino acids, right? There are the polar amino acids, hydrophilic. There are and then some people will parse this in a little bit of a different way. Some people would tell you there are only 3 flavors. They'd say the 3rd flavor are the charged amino acids. I'm more of the school of thought of saying they're not, let's be more specific, they're not just charged. One category are the positively charged, and another category are the negatively charged. Okay? So we got, in my way of thinking, we've got 4 different kinds of R groups, 4 different kinds of amino acids. Some that impart a little bit of hydrophobicity to a protein, some that impart a little bit of water solubility and maybe a whiff of chemical reactivity, some that are intensely negatively charged and therefore probably very reactive or very interested in making ionic bonds at the very least with other things, and some that are some amino acids that we can use to impart a positive charge to a small portion of a polypeptide strung together. So we've got these 3 or 4 different kinds of amino acids. And you can see here, this particular figure from your textbook shows you the non-polar side chains, the hydrophobic ones. They're all grouped together up here, and they're shown with yellow for where their R groups are. The bluish, is that what we should call this, right? This bluish color here is just the core part of all amino acids. See that amine group, see the central carbon, see the hydrogen, see the carboxyl group. Ditto for all of these. That's what all of these blue things are. But for the hydrophobic ones, they're trying to have you paint here with yellow. When you have a hydrophobic R group, it's yellow. When you have a hydrophilic R group, they're using, give me a color here, guys. Teal. It's teal or green, right? Something like that. So pick whichever you're more comfortable. If you want this to be a fancy sort of class, we'll call it teal. If you want it to be sort of a, down-to-earth kind of class, that's green. Whatever is good for you, right? But so they're trying to show you this different hue that comes from these amino acids. Over here, you've got the negatively charged one, salmon or pink, I suppose, depending if you want to be fancy or hoity-toity, or I'm sorry, if you want to be down-to-earth about this. And then, boy, if this thing was teal, what are we going to call that? Let's just call it blue. Right? So you can see that the textbook is using this same sort of theme of different colors that you could use to make different molecules. Now again, I don't think you can read this slide from where you're sitting. If you can, you've got really good eyesight. Good job. You know, keep it up, whatever you're doing. Eat more carrots, right? Keep your eyesight strong. But here, the next couple slides lets me zoom in on them. So let's just focus our attention at the start at the hydrophobic guys, the R groups that are hydrophobic. Remember, all amino acids have that same purplish stuff, right? The amino group, the central carbon, a hydrogen dangling off of it, and a carboxyl group. So there's not a lot of action going on down in that part of the molecule. The differences from one amino acid to another are what's going on with that R group, okay, that reactive group. They're side chains, sometimes they're called. There are 1, 2, 3, 4, 5, 6, 7, 8, 9 different amino acids that fit into this hydrophobic category. Okay? Pretty straightforward. Each of those 9 different amino acids has its own name. People have taken the time, they see them often enough, they play an important enough role that they get a name. They have names that all end in -ene, all amino acids end in -ene. But you can see the names. Do I need to read them to you? The first one on the list there is glycine. You got alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan. Okay, that one doesn't end in -ene. Sorry about that. That's a weirdie. I mean, look at the structure anyway. That's it. But and then you got proline. And for shorthand purposes, it becomes cumbersome to write out glycine all the time. And so it's quite common that you'll see it, all these amino acids have very standard 3-letter abbreviations, and they even have standard 1-letter abbreviations. Because in a little bit, I'm going to try to impress upon you how the sequence of amino acids in a protein is really important. Okay, so, you know, you could, for the sequence, you could say I've got a glycine followed by an alanine, followed by a glycine, followed by a tryptophan. Or if you wanted to write that out in shorthand, you could just write GAGW, right? And to somebody in the know, they would immediately be able to translate that into what I said in a more cumbersome way, glycine, alanine, glycine, tryptophan. Okay? So you can see the full names of all of these 9 different hydrophobic amino acids. You can see that there are 3-letter abbreviations, make pretty good sense given the names. You can see even that the 1-letter abbreviations make pretty good sense, with some interesting exceptions like the weirdie tryptophan, right? Who would have thought you'd abbreviate tryptophan with a W, right? There actually is a method, a reason behind why it's abbreviated W. Part of it is because there are other amino acids whose first letter is T, right? So you can't have everything that starts with T be labeled T, but there is a methodology to the nomenclature. I got real good news for you though, right? I told you I'm not going to make you memorize these structures. If you don't memorize the structures, there's no point in memorizing the names. If you want to take the time to associate the structure with the names, go for it. I think that's a swell idea. But I'm not going to quiz you on that for the test that's coming up 2 weeks from today. Okay? What I do want you to be able to do is if I were to show you the structure of an R group, not even the whole amino acid, if I just showed you an R group and I asked you, is an amino acid with this R group going to be hydrophobic, hydrophilic, positively charged, negatively charged, or I don't know, I'll make something up, zwitterionic. It's a chemistry term, right? You know, which is it, right? And if I showed you the R group, I've said the R group I'm talking about is an H, nothing but a hydrogen. I hope you'll be able to trot out, play this out. Gee, what's the H doing? It's got a covalent bond with a carbon, similar electronegativity, similar affinity for electrons. Gee, that's going to be a nonpolar covalent bond. Nonpolar covalent bond, there's nothing for water to sink its teeth into. Okay, that's hydrophobic. Right? I hope it plays out a little bit faster than that as you crank through that wheel. But that's what I hope is going to be the wheels that get turned when I ask you that question. So if I show you an R group and that R group is just an H, you can feel high and mighty and say, oh, we're talking about glycine. I'm not going to ask you if it's glycine. It is. But I hope the salient feature here, the most important thing is, oh, this is an amino acid. If that's the R group, this is going to be a little patch of hydrophobicity, water afraidedness, right, in English as opposed to the Latin. And check out the rest of these other hydrophobic R groups. We got a word for this, CH3, that's a methyl group. Methyl group, gee, hydrophobic, right? That's the word that I hope springs to mind. What's next down the line? Valine. Oh my gosh, that guy has 2 methyl groups and, you know, the beginnings of a third methyl group. I mean, that's just got, I hope that's screaming hydrophobic to you. Move on down. Leucine, it's getting worse, right? There's even more methyl groups there. It's just, you know, hydrophobic is hydrophobic, right? But this guy is, you know, flaming hydrophobic. He's like really showing the world, I'm hydrophobic, all right? Further on down, isoleucine, that guy's pretty seriously hydrophobic too. That's a big hydrophobic, right? And I want to emphasize big. You know, let's say you're designing a molecule. Let's say you're designing a protein. You're a protein engineer, and you wanted to dial up a hydrophobic patch in this protein that you're making. Any one of these 9 hydrophobic amino acids would do. And in fact, they're pretty much interchangeable in that sense. But let's say you wanted to have something bulky and hydrophobic, right? What do? You go for some of the ones further down on the list, right? Let's say you want to have something that's just hydrophobic, yeah, but doesn't really get in the way of other stuff, doesn't poke out too much. Hey, we got something for that. That's going to be glycine, or maybe alanine, or maybe valine, right? And so nature, when it's painting with this palette, it has 20 different amino acids. It's got essentially 4 different color schemes, but within each of those 4 different color schemes, the principal difference from one of the amino acids to another is really just how bulky it is, how big it is. There are going to be times when it's going to come in handy to have something big fill in that space, and other times you want something small and sometimes something in between. Functionally speaking, most of these amino acids can be interchanged. I'll talk with you about that a little bit more in just a couple of minutes when we talk about amino acid sequences. They pretty much all do the same job, but there's the opportunity to do it with a big molecule or a small one. That's what we've got going on here. Now, for all of these structures, there is pretty much nothing to see but carbon to carbon and carbon to hydrogen bonds. The very definition of nonpolar covalent bonds, hydrophobic bonds. You've noticed, I'm sure, there are a couple of sort of oddball exceptions. There's a sulfur here in methionine. Sulfur's affinity for electrons is very similar to that of carbons and hydrogens. Doesn't really do much here in terms of making it hydrophilic. Right, so methionine stands out a bit. It's the only amino acid that has sulfur in it. We'll talk a little bit more. I've already told you a little bit about why that's special. It lets you make a special kind of bond called a disulfide bond. We'll get to that in more detail in a bit. So methionine, a little bit quirky, but still hydrophobic. And over here, tryptophan, yeah, there is that nitrogen there. But that nitrogen is pretty well embedded in 2 rings, structures full of carbons, full of hydrogens. All in all, tryptophan, more than anything, is big. Okay, that's like the biggest R group you are going to see. It is literally bulky, it's big. It actually is on a par with the size of the rest of the amino acid. Right? Most of these R groups are little add-on things tacked on to that core structure. Tryptophan's R group gives a good run for the money in terms of the size of the amino acid itself. So tryptophan is primarily big, and it's okay, maybe it's not quite as hydrophobic as the rest, but it's plenty hydrophobic enough to be in the hydrophobic category. All right? So those are the hydrophobic ones. Again, don't get freaked out right now about the structures, okay? I'm not that worried about you memorizing the structures. I'm not even that worried about you memorizing the names of the 20 different amino acids. I don't want to discourage you from doing that, I think it's a fun exercise. Okay, it's an important exercise. It's something that would serve you well down the line, but I'm not going to ask you about that sort of thing 2 weeks from today on the first test. Instead, I'm going to challenge you with the structures, the R group structures, and I'm going to ask you what I think is really important about them, what it is they do in terms of the functionality of the molecules that have them. All right, so be able to look at those structures, figure out on the fly, is this hydrophobic, hydrophilic, positively charged, or negatively charged? Of course, if you memorize the structures, you won't have to figure it out on the fly. So I'm good with that. I'd rather you know how to do it on the fly as opposed to memorizing it. But let's move on. So we've got those 9 different hydrophobic R groups that give rise to 9 amino acids that we characterize, we say are hydrophobic amino acids. Here we're looking at 6 polar R groups, or 6 polar side chains, hydrophilic as opposed to hydrophobic. These amino acids made with these R groups, or these side chains have a little bit of hydrophilicity associated with them. For the hydrophobic ones, it's essentially nothing but carbons and hydrogens all day long. For these guys, lots of carbons and hydrogens, but the first one on the list here, serine, we got a word for a carbon compound that has that dangling off of it. That's hydroxyl group. We call those things alcohols, but the hydroxyl group there, that's different than what we had seen on the previous slide. There is going to be a partial separation of charge. There's going to be a little bit of negative charge on the oxygen, a little bit of positive charge on the hydrogen, and maybe even a little bit of positive charge on that carbon that's part of the structure. But in a word, there's a basis for interaction with water, whereas we hadn't seen that before. And just as with the hydrophobic amino acids, the principal difference aside from that is the size. Some of these guys are relatively small, some of these guys are huge. All right, that big ring, again, the molecular weight of the R group is comparable to the molecular weight of the rest of the amino acids. So you go everywhere, all the way along the way here, from being relatively small and diminutive to being pretty darn big. And you got a couple of choices in between. The hydrophilic guys have hydroxyl groups, or they'll have here, you can see a sulfhydryl group, like what we have in cysteine, and you'll find asparagine's got, this is clearly, there's no full charge associated with either of these, but there's going to be separation of charge, a little bit of positive here, a little bit of negative there, and the same is going to be true over here for glutamine. All right? So, if I confront you 2 weeks from today with a picture of an R group from one of this category, what you'll do is your attention will be drawn to the hydroxyl group, maybe it'll be drawn to the sulfhydryl group, maybe it'll be drawn to the amino and carbonyl thing that's going on with the last two, and you'll pretty well be able to crank the wheels and say, this is going to be water soluble, this is going to be hydrophilic, this is one of the polar R groups. All right? Then let's get to the guys with the full charge. Again, some people will tell you there are only 3 categories. I like to talk about 4. I split one of the categories up into 2. These are not just charged. I think we can easily distinguish between those that are negatively charged and those that are positively charged. There are only 2 amino acids that have consistently full negative charges associated with them. When you find an aspartic acid or a glutamic acid as part of the structure of a protein, you should be expecting there will be a full negative charge there. Okay, so we've got 2 negatively charged amino acids. Why are they negatively charged? Well, for goodness sakes, these are the only 2 R groups that have carboxyl groups associated with them. All right? Carboxyl groups, negative charge. Got 2 that get a negative charge, and then you got 3, lysine, arginine, and histidine, that have a positive charge. Why do they have a positive charge? Because they've got those amino groups dangling off of them. Arginine and histidine, in some sense, have two. So you got positive charge associated with them. The difference between aspartic acid and glutamic acid, essentially size. Right? Once you've gotten past the big point that they're negatively charged, the only other thing that matters here, if you want something that's negatively charged and bulky, you plug in glutamic acid. If you want something that's negatively charged and not quite so bulky, plug in aspartic acid. For the positively charged guys, you can see you get a little bit more, they're pretty consistently big. Some of them have subtly different, a little bit bigger, a little bit more localized positive charge, like arginine. It's just like waving positively charged stuff around. This one's not waving quite so much. This one's held a little bit closer in, but nonetheless positively charged. All right. One more time. We want to paint some pictures. I want to be able to build some molecules that can do remarkable things, catalyze very specific chemical reactions, and only one chemical reaction. The way that I get that done in a living system is with a protein. That particular biopolymer, I have to string together amino acids in a particular sequence such that the protein brings to bear, in just the right orientation, a certain set of reactive groups, these R groups. It's all about bringing to bear that little spot on the surface of the protein, typically, just the right combination of negatively charged things, positively charged things, hydrophobic things, to coax in a substrate that you can then convert into a product, right? Catalysis. That's what enzymes are all about. That's what proteins are all about. And we do that by stringing together different combinations of these 20 different amino acids with their 4 basic different kind of colors associated with them. Okay? Those are the structures of the amino acids. That's what makes them different. Let's talk about stringing them together, and then let's talk about what you can do once you've strung them together. That's, I think, really cool. So again, talking here about a polymer. Like all polymers, we've got a monomer. The relevant monomer for a protein is an amino acid. You link together one monomer, one amino acid, to another monomer, another amino acid, through condensation reactions, just like is shown in this particular image. Okay, so we've got an amino acid with this hydrophobic R group and an amino acid with this hydrophilic R group. Anybody want to impress me and shout out the names? Feel free, right? I don't care. I'm really focused on the fact that this is hydrophobic and this is hydrophilic. And you can add a new amino acid to this existing chain through a condensation reaction. And that involves the making of a covalent bond between the carboxylic acid of one amino acid with the amino group of an incoming amino acid. For proteins, the making of this bond only happens in this way. You don't ever tack on more amino acids at this end of the molecule. You always tack them onto the carboxyl end with an incoming amino group, always. Okay, so this is a condensation reaction, and you make a covalent bond. You can see they're showing it to you here with the arrow in blue, between that carboxyl group and the amino group, and you kick out a water molecule. That's why we call it a condensation reaction. Do you recall, probably said it too quickly on Tuesday, the bond between one monosaccharide and the next monosaccharide has a fancy name. It's called a glycosidic bond. Okay? I don't think there is an equivalent word for lipids, right? But for proteins, the bond that connects one amino acid to another amino acid, that's called a peptide bond. Okay? The only time you'll ever hear peptide bond is when you're talking about the bond that connects one amino acid to another amino acid. So down here in this part of the picture, there are actually 2 peptide bonds. That's a peptide bond, and that's a peptide bond. And if you added on a third amino acid, you'd do it at this end, and you'd have a fourth peptide bond over there. Okay? When you string together these amino acids, you can fold up this long chain of amino acids to give rise to things that look at a molecular level like what you see up on this slide. A real big part of bioinformatics is taking that information that we can determine pretty readily these days about the order in which amino acids got linked together, and then taking that information and predicting what sort of structure it will fold up into. It turns out to be a really difficult problem that hasn't quite been solved yet. But boy, the wondrous things that will happen once we figure out how it is that proteins fold up, when we can predict what the structure of a protein will be, it will allow us to design proteins that can do really cool stuff. All right, just whatever you want to have done, we can probably make a protein to do it. If only we can figure that last part of the puzzle out, how they actually fold up, and we can predict with confidence this is the structure it will have. What sort of cool stuff am I talking about? I mean, anything you could imagine, we could probably design a protein to do. Did you guys know that in seawater, for every liter of seawater, there's about 1/10 of an ounce of gold dissolved in it? Maybe less, about 100th or 1,000th of an ounce of gold. But there's lots of gold. There's lots of liters of seawater. It should be pretty easy, once you know how to fold up proteins, to design a protein that binds to gold dissolved in seawater, precipitates it out, and leaves you little gold flakes at the bottom of the tube when you've been playing with the seawater. I mean, you want to make gold? There you go. That's how you do it. You want to make a nanotube that circles the globe? And we could do that if we knew how to predict how these proteins fold up on themselves. We're close. We're not quite there yet. But ultimately, again, what happens in nature with these proteins is they fold up into these neat structures. Here is a ribbon model. I don't need to tell you that. But here is a space-filling model of a protein. And it doesn't take a very well-trained eye to be able to recognize where the action on most proteins has taken place. It's usually, there's like a neon sign almost after you've looked at them a little bit. The action for this particular protein is taking place right in here, right? And what's going to happen is all the rest of this structure, all the rest of these amino acids and the atoms that they bring with them are there primarily just to make sure that this part of the protein folds up in just the right way. Get a couple positive charges. You get maybe a negative charge hanging out in just the right three-dimensional conformation to be able to entice in a substrate. Something that's going to get reacted. This is going to act as a catalyst. Something you want to be able to pull in and get nice and cozy. Molecule fits like a lock and a key. It's often the way that it's described. You get a molecule that can fit in, and that, in this molecule, this is called the active site. That's where the chemical activity, where the catalysis is likely to be taking place. Active sites usually hit you in the face when you're looking at the three-dimensional structure of a protein. I mean, they leap out at you. It's like, oh, yeah, something's going to be happening over here. What's going on? Excuse me, what's going on over here? Probably not too much. What's going on over here? I don't know, probably not that big a deal. What's going on over here? Oh my gosh, that's a pretty big pocket. There's probably something really important that's happening there. So here they call it a groove. That groove is associated with the enzyme's active site. How do we get to those structures? Well, again, you do it with the specific sequence of amino acids. Our genes, we're going to talk about nucleic acids next, but our genes spell out for our cells the specific order in which amino acids should be strung together to make a protein. I'll steal a little thunder from what I'll be telling you in a few minutes. An organism like you and I, we've got in the ballpark of 30,000 genes. Each of those genes gives rise to at least one protein. And any of them give rise to more than one. But that's a subtle point, and that's for later in the course. But we can make on the order to be a happy, healthy, functioning human being. At some point during the course of your life, you probably have to be able to muster up the ability to make on the order of 250,000 different proteins. Your genes are what codes for those proteins. One protein differs from another protein simply based on the sequence with which its amino acids get linked together with these peptide bonds to make these polymers. I'm tempted to tell you I know the name of this protein, and it helps me that it's written out right there, right? Because the one I was going to guess isn't the right thing. You know, you want to hear the kind of torture you go through to get a PhD? And there's some classes you have to take early on. I got my PhD at Penn State. My PhD is in biochemistry, strictly speaking. That's what the diploma says, biochemistry. Biochemists are all about proteins and protein structures. That's what it's all about. In a biochemistry class I took as a graduate student, the jerk of a professor made me memorize the primary sequence, the order in which the amino acids were linked together for the protein insulin. What kind of useless knowledge is that, right? It's 51 amino acids long. That's still seared in my memory, but I had to spend hours memorizing the specific order, coming up with clever mnemonic devices for the order in which those amino acids get strung together.
You still know it?
I don't think I knew it for the test, let alone now. You know what I remember? It started with methionine. Every protein, except for this one apparently, starts with methionine. Yeah, so methionine a pretty good bet for the first amino acid for most proteins. But the nice thing about having done that stupid exercise is that it really strikes home to you that if you're off by one amino acid, that's not insulin anymore. And it probably won't work as insulin, or at least won't work as well as the version of insulin that didn't have that amino acid off. Every protein, 250,000 some different proteins that we make, every protein has a different primary amino acid sequence. That's what makes a protein different. And that primary structure, you can see that's what this slide is all about. That primary structure is simply the linear order in which you'll string together all of these 127 different amino acids. Now, biochemists will they'll tell you that this is an important distinction, the amino end as opposed to the carboxyl end. You know, it's important. It's kind of subtle. What's particularly important is that the amino end, right, this is where the amino group is still hanging free. That's the first amino acid in this polymer. So you could think of it as the beginning of the protein. The last amino acid to get incorporated in is this one, glutamic acid. It's still got its carboxyl groups waving out in the wind, ready for another amino acid to be added on if you were to extend this polymer any further. So there's a beginning to a protein and an end. The first amino acids that get linked together are at the amino end. The last to get added on are at the carboxyl end. But aside from that, beginning and end, it's all just about which of those 20 different amino acids you strung together. Okay? I think this says something important about me and my way of thinking about stuff. I know, bam, I can tell you how many amino acids are. Don't hesitate, don't flinch. How many different letters are there in our alphabet? I wrestle with this. Is it 26 now? All right. So for a while, I was telling this class, not this group, but other years, we got 24 letters that we spell out all our words with. So not as good with the English alphabet as I am with amino acids, right? But in the same way that you can string together a whole wide variety of different words that convey a whole bunch of different meanings by different arrangements of our 26 letters, you can string together a whole bunch of different proteins with different arrangements and different lengths of these 20 different amino acids. All right? That string, that linear sequence of amino acids linked together, we'll call the primary structure. It's usually one with a superscript 0 or a degree symbol next to it. That's the primary structure. Now, if there's a primary structure, it probably follows that there's a secondary structure. And in fact, there's a tertiary and even a quaternary structure associated with proteins. The primary structure is, again, the sequence with which, simply, the sequence with which the amino acids get strung together. The secondary structure is there are certain three-dimensional conformations that particular strings of amino acids tend to fold up into. There's essentially three different kinds of secondary structure. There are alpha helices, there are beta sheets, and there's stuff that's neither an alpha helix or a beta sheet. All right? Just sort of random stuff. An alpha helix. You know, I love that this is a vintage classroom because I get a prop to use. All right? You guys remember what telephone cords looked like? All right? Back in the old days? That cord is an alpha helix. Okay? You can come down and touch it after class if you like, because I know that not many people have seen those anymore. But telephone cords, that's the perfect example of an alpha helix. Right? And so certain strings, certain primary sequences of amino acids simply have a tendency to fold up in a particular way that causes them to have that kind of coil, an alpha helix. Right? It's as simple as that. I told you bioinformaticians are really big into being able to predict from the primary sequence what sort of secondary structures will arise. We're pretty good at being able to recognize features, characteristics of the amino acids in a primary sequence that make it likely for them to fold up in an alpha helix. Right? There's a lot of theory and complicated math that's involved, but we can predict with pretty good confidence if a string of amino acids will fold up in an alpha helix. And by the same token, an alternative to an alpha helix is a beta sheet. We can make pretty good predictions about beta sheets as well. Right? Beta sheets. See, you guys learn way more about me than you probably should in a class like this. You know, when I was in high school, my daughter's a freshman in high school this year. She plays bassoon, right? So she's really quite good at bassoon. You know, I think that my wife and I foisted upon her a reasonably good choice of instrument. When I was in high school, the instrument that my parents had me learn was an accordion, right? Not just any accordion, but a button box accordion. So you can imagine what a cool high school student I was walking around with my accordion. I should have brought one of my accordions in with me today to give you a little song. It would have made this more memorable. But the pleats, right, and the accordion, that's a beta sheet, right? That's when you guys have probably encountered beta sheets in your day-to-day life, like when you go back home at the end of the day and you break out your accordion to unwind a little bit, all right? So, helices, beta sheets, here they're calling it a beta strand, or neither. It's pretty much your three choices for secondary structures. But the primary structure dictates to a very large extent if you're going to form one of these two or three different kinds of secondary structure. Okay? The secondary structures, in turn, cause the long polymer of the protein to contort and fold up in a certain way that gives rise to a more complicated three-dimensional structure. That's what we're talking about for the tertiary structure. It's the overall sort of three-dimensional configuration of the protein that's governed ultimately by its primary structure, but then guided along by its secondary structure, such that those alpha helices and beta sheets bend up and fold up in a certain way such that you get a particular tertiary structure. We're still not very good at going from secondary to tertiary structures in our models, right? But nature does it really well all the time. So there are rules. We just got to figure out what they are still. And then the quaternary structure idea is simply this. Most proteins don't act onto themselves. They need to be physically associated with other proteins, sometimes other copies of the same protein, sometimes other proteins altogether. Those interactions between fully folded proteins, those are called quaternary structures. Okay? So these are the 3 or 4 different levels at which we can talk about the structure of a protein. Let's see if we can look at them a little bit more closely. The alpha helix, again, is just that telephone cord. The beta sheets, or the beta pleated sheet, those can actually interact with each other. One beta sheet relative to another can form a fairly ribbon-like flat structure as part of the protein. You can, they're showing you here with this ribbon model, they're trying to draw your attention. This is an alpha helix. There's a beta sheet here, another one that's running in the opposite direction there. Again, notice how the beta sheets are often found near each other. There's 4 of them that are probably all interacting with each other in a certain way. Here we've got another 4 beta sheets interacting there. This one's only got the 1 alpha helix, and you got the other stuff that is neither. Again, we can predict with pretty good confidence if an amino acid sequence will fold up in an alpha helix or a beta sheet, and we can get ground truth as to whether or not that's what's really happening at the molecular level in a cell. That ground truth comes typically from X-ray crystal structures, X-ray crystallography. So you can zap these crystals of these proteins with X-rays, get a diffraction pattern, and then use fancy math, Fourier transforms and such to be able to predict back the physical location of the atoms that gave rise to that scattering of the X-rays. Bottom line is that you can tell what the structure actually was, and then you can look back and compare how well your prediction was to what the structure is. And so, you know, here's a particular protein. This protein is called transerythrin, and it folds up in this particular way. And transerythrin, it turns out, apparently works as a tetramer. 4 copies of the protein have to come together to be able to make the functioning version of transerythrin. There must be something important going on here, is all I can tell you. Right? Again, those active sites where the business end of the molecule is usually jump out at you pretty clearly once you've got those tertiary and quaternary structures to work with. All right. There was a term I wanted to mention to you. Well, here's hemoglobin. Hemoglobin is a tetramer. There are 2 copies of the beta protein, beta globin, and 2 copies of the alpha globin that come together to make hemoglobin. Heme is an important part of hemoglobin. It gets stuck into the globins here in those 4 spots, and heme has an iron in it that can be used to interact and have a relationship with oxygen. So that's what's carrying oxygen in our bloods. There was a word I wanted to just toss out at you here, a phrase. It's called hydrophobic collapse. Okay? When you're thinking about how does a protein fold up, right? Part of it is this alpha helix stuff, part of it is the beta sheet stuff. But here's another thing to bear in mind. Many of the amino acids, 9 of them, are hydrophobic. Right? If you have stretches of hydrophobic amino acids, one after another, where do those guys like to hang out, right, when you're folding up this protein? They want to get as far away from the surface of the protein as possible, because the surface, the world in which proteins are living, is a very watery environment. These hydrophobic R groups want to get as far away from water as they can, so they tend to fold in on themselves, and they find other little patches of hydrophobic stretches of amino acids, and hence this idea of hydrophobic collapse. The interior of the tertiary and quaternary structure of a protein, the interior is typically where those hydrophobic R group amino acids are hanging out. And on the exterior, that's where you display and you flaunt your hydrophilic and your charged amino acids. And it's at the active site of the enzyme that's not only on the surface, but where the catalysis is taking place. There you usually have a couple really strongly charged R groups floating around to help facilitate some catalysis. So again, just this idea of hydrophobic collapse, another important driving force in folding up of proteins is you try to hide the hydrophobic amino acids buried into the middle of the protein's structure, and you leave on the outside all the things that are a little bit hydrophilic or very hydrophilic, the charged amino acids. All right, so there's my spiel about proteins. Let's get to work on nucleic acids. Best part of all for these sorts of things. Nucleic acids are a polymer, just as proteins are a polymer. We'll be beating this slide to death in another couple of weeks. But nucleic acids, if proteins are what gets stuff done, let me put that as a question. Proteins are what gets stuff done for cells. That's what those biopolymers are used for. They're used to catalyze chemical reactions. They also play some important structural roles. Okay, so proteins are what gets stuff done. They're literally the muscle of cells. Nucleic acids, in contrast, are the blueprints. It's where all the instructions are stored that are used to make the proteins that get the stuff done. So you could think of the nucleic acid sort of as the brains of the outfit. It's probably better to think of it as the blueprint for the rest of the cell. And the way that nucleic acids, well, and that sets up this scheme then where you store information in DNA, you transcribe it into a transient molecule called RNA, another kind of nucleic acid. And then the information in that transient molecule, the RNA, gets used to string together amino acids to make proteins. And that's what's playing out here in this particular slide. But the information gets stored in the sequence with which different nucleic acids get strung together to make this biopolymer we call DNA. 7 different functional groups that come up time and again. 20 different amino acid R groups. Turns out only 4, we can quibble and say 5, but for now let's just focus on 4. Only 4 different nucleic acids that we really worry about, at least in the context of DNA. Right? Only 4 building blocks, only 4 letters. You know, that translates to something fairly directly. It would be unreasonable to expect DNA to do very much in the way of catalysis, right? It's going to be hard for DNA to get much done because there's only so much diversity, only so much variety you get from 4 different nucleic acids. You have, a relatively constrained palette to work with. There's not that much difference in terms of chemical reactivity, especially in contrast to something like proteins, where you got 20 different colors that you might paint with. So nucleic acids, you only got the 4 to worry about, and yet what you can do with those 4 is pretty remarkable. You string them together, they're each a monomer, they get linked together through condensation reactions like all these other biopolymers. You pull a water molecule out, you make what's called a phosphodiester bond. That's the name of the bond between the 2 polymers. But you use condensation reactions to string these guys together. You use hydrolysis reactions to break them apart. But it is the sequence with which you string together those 4 different nucleotides. If you like, those 4 different letters, it's the sequence that determines all the rest. It's the blueprints. And again, I've said this earlier today, you can make monstrously long polymers of nucleic acids, hundreds of millions of nucleotides long, storing enormous amounts of information. About 15 years ago now, a private enterprise endeavored to patent the human genome. Right? They were involved, they had been involved with sequencing, determining the order of all of our 3 billion nucleotides, how they're arranged, 3 billion nucleotides. And they tried to get a patent on that. The patent application that they submitted to the U.S. Patent and Trademark Office was 26 CDs. Right? You guys have a sense for how much information you can store on a CD? It's like anybody here ever touch an encyclopedia? Another sort of historical and oddity here. There used to be these books, right, that would have all these little stories spelled out, and now we have Google, right, that sort of supplanted the entire need of encyclopedia. But you could put an entire set of encyclopedias on a CD, right? It took 26 of those to spell out the 3 billion letters of the human genome. That's a big genome. All right, so we got this big genome, all of this information. In there is everything that you need, not everything, but an awful lot of what you need to be able to make a working, functioning human being. Right? And it just comes down to the specific order in which those 4 different nucleotides get linked together. Our DNA alphabet is pretty constrained. You only got 4 letters. 26 letters, apparently, in the English alphabet. All right? The upshot of that is that we can have relatively short words that convey some very specific meanings. D-O-G, right, is one combination. G-O-D, different combination, relatively short, very different sort of ramifications associated with those 2 different arrangements. When you only got 4 letters to work with, your words simply have to be longer, right? If I only had 4 letters in the English alphabet, I could work with that. It's just my words are all going to be really long. DNA spells stuff out, genes, it spells stuff out into relatively long words as a result of the fact that it's only got 4 letters in its lexicon, in its alphabet to work with. Okay? And that's part of the reason why we have such a monstrously large genome of 3 billion nucleotides. But let me point you to some of the key features of a nucleotide. So just like amino acids, there's this sort of core part of all nucleotides, and then there's this one little variable part. The core part of a nucleotide is a 5-carbon sugar. In DNA, it's deoxyribose, meaning that one of the oxygens, one of the hydroxyl groups, is missing from the 5-carbon sugar. In RNA, it's ribonucleic acid because the hydroxyl group is still there. But you have this sort of core central 5-carbon sugar, to which is attached a phosphate group, and also to which is attached a nitrogenous base. Now, all nucleotides have the same basic structure, a phosphate group and 5-carbon sugar. What differs from one nucleotide to another is what the nitrogenous base is. So you can see that for DNA, we've got cytosine, thymine, adenine, and guanine. Okay? Those are the 4. In RNA, in place of thymine, we use uracil. Now, once again, I don't expect you guys to commit to memory the structures of these 5 different nitrogenous bases. But I hope you appreciate that there are 5, 4 when you're talking about DNA, G, A, T, and C, 4 when you're talking about RNA, G, A, U, and C. Okay? So there's 4 where you're talking about U and T are interchangeable. U is used in RNA, T is used in DNA. I want you to understand that there are 4. It would probably be a good idea if you appreciated that they have 1-letter abbreviations, right? All the cool molecular biologists, no cool molecular biologist talks about guanine. They talk about G, right? Guanine is almost an artifact these days. It's just like nobody talks about guanine. They talk about G. Nobody talks about adenine. They talk about A. Right? So it may as well G, A, T, C are the 4 DNA nucleotides or nitrogenous bases. Swap out the T and plug in U for uracil for the RNA nitrogenous bases. And there's one more other thing about these structures that I hope you can pull away for this class. We can divide nucleotides or nitrogenous bases into 2 categories. There are the pyrimidines and the purines. So the pyrimidines have just 1 ring. The purines have 2 rings. Okay? Guess what? The purines are bigger. The pyrimidines are smaller. And I'll tell you very simply, I have to do this all the time when I'm talking about purines and pyrimidines. I have to remember it's the exact opposite of what you would think. The thing that's the simpler structure has the longer name. Right? I use this all the time. Pyrimidines have 1 ring, purines have 2 rings. That ends up having some important implications for the way that double-stranded DNA gets put together. I'll talk with you about that just a little bit. All right, so there are two different kinds of nitrogenous bases in a broad sense. There are the ones with one ring, the pyrimidines. There are the ones with two rings. If I were to show you a structure like this on an exam and ask you, is this a pyrimidine or a purine or an amino acid or a monosaccharide, I hope you'd be able to conjure up that's a purine. Okay? I showed you this. Is this an amino acid? Is it a carbohydrate? Is it a lipid? Or is it a pyrimidine or a purine? That's what a pyrimidine looks like. So that's the level at which I hope you'll have an operational knowledge of what's going on here. You don't have to know that this particular pyrimidine is cytosine as opposed to thymine, but notice that it is possible to, there's that methyl group there that's not there, there's the amino group there that's not there, right? There are differences between them, it's just you don't have to worry about them right now. So, again, the way that information gets stored in DNA is the specific sequence with which nucleotides get strung together by way of phosphodiester linkages to make a long polymer. That's the way the information gets stored. GAGT conveys a different nugget of meaning than GTGA, in the same way that DOG means something different than GOD. It's all about the sequence with which these nucleotides get strung together. And those, in turn, are used by cells to determine the order in which amino acids get strung together. There's a three-letter code. 3 nucleotides correspond to one specific amino acid. Right? So you can spell out which amino acid to put in by different combinations of those nucleotides. But that's how information is stored in DNA. And I think you guys all know, it's a stereotypical kind of image for molecular biology, that DNA is not just one strand. We tend to think of DNA as a double-stranded molecule, even more specifically as a double helix. What's going on with the two strands of DNA is simply this. On one strand, you'll have a specific sequence of nucleotides. Let's say it's GAT. Okay? On another strand, complementary to that, it turns out that G's get along well. They fit comfortably with C's. So if you've got a G on one strand, on the other strand there's going to be a C. If you've got an A on one strand, on the other strand there's going to be a T. If you've got a T on one strand, on the other strand there's going to be an A. If you've got a C on one strand, on the other strand there's going to be a G, right? The two strands of a DNA molecule are complementary to each other. It's sort of a backup system, an intrinsic backup system. If something took a bite out of one of your strands, you'd know what information to plug in simply by looking at the nucleotides that are on the other strand. It also gives rise to a neat way to replicate DNA. You can separate the two strands and make two new double-stranded molecules using the information that was on those first two strands that you separated. So DNA is a double-stranded molecule typically, and the information on the two strands is redundant. The strands are complementary to each other. If you have a G on one side, you have a C on the other. If you have a T on one side, you have an A on the other, and both of those permutations, C to G, A to T. Okay? So that's how information is being stored. And it is possible for nucleotides to fold up based on, that's called base pairing. So you can fold up nucleotides in particular ways. This is what a tRNA molecule looks like. But that's what I wanted to tell you guys about nucleic acids, at least for now. We'll get into them more in more detail later. This slide here, a PowerPoint presentation later, is just a one-slide summary of all the different polymers, what they do, what their bonds are called, what their monomers are, and stuff like that. All right? So have a great weekend. See you guys all on Tuesday.